Yes, so I want to use this time to actually give you some brief lecture on the glucose metabolism. So we say that whenever we metabolize glucose, if you remember respiration, whenever a cell undergoes respiration, how many molecules of ATP are produced?
38.
38, okay. We're not going into details to go and know how all those are produced, but I'll just take you through the steps. What you need for the exam is 38 molecules of, when one molecule of glucose is hydrolyzed or oxidized, we generate 38 molecules of ATP. Now you know that the glucose metabolism actually begins with what? When we are breaking glucose for energy, it begins with glycolysis, right? Then from there,
Yes, Doc.
you move towards the Krebs cycle. Then from there, you move towards oxidative phosphorylation. Do you remember that oxidative phosphorylation actually came in June MCQ? Do you remember? Or to date, you have still not gone through the MCQs? No. So these are the steps, okay? So the first step is actually what? The glycolysis. And remember that where in the cell does glycolysis take place?
Cytoplasm.
In the cytoplasm. Where in the cell does the Krebs cycle take place?
Mitochondria.
How about oxidative phosphorylation?
Also mitochondria.
So remember that the mitochondrion is what? The powerhouse of the cell from GHS. Hey, please hold on. Good. So please take note of these facts. Glycolysis occurs where? In the cytoplasm, Krebs cycle, mitochondria. And the last one, oxidative phosphorylation, mitochondria, okay?
Yes, Doc.
Now, remember that we have done glycolysis, okay? We just took note of the important points. Okay, what are the steps? Can you quickly take us through?
Of glycolysis, the glycolysis.
Yeah, the steps, yeah.
So
Give us the mnemonic,
glucose,
please.
the mnemonic, great grandmother fears fighting great granddad because 3-2
Great
punches.
grand, please, you are moving fast too. Great grand, mom.
Yes.
Fears fighting. Great
granddad.
Great grand, right? Dad.
Yes, Doc.
Because
32 punches
32 punches paralyzes, isn't it?
Yes, Doc.
Okay. So the great stands for what? Glucose, right?
Yes, Doc.
So glucose is acted upon by hexokinase or glucokinase to form what?
Glucose 6-phosphate.
Thank you. And then glucose 6-phosphate is isomerized, right? To form what?
Yes.
It's isomer, right? So that is what? Fructose 6-phosphate. So we are just changing the molecules, the arrangement. Fructose 6-phosphate. Then phytin. You add another phosphate to this guy, isn't it? Then we have what?
Yes.
Fructose 1,6 bisphosphate or diphosphate, isn't it?
Yes, Doc.
Then the fructose 6-phosphate is actually divided into what? 2 molecules. Remember that glucose is, how many carbons does glucose have?
Six.
This one is not, is integrated sign. So how many carbons? Yes.
6 carbons.
Yes, and so has fructose, isn't it?
Yes, Doc.
So if you are dividing fructose 6-phosphate into two halves, it means that each half will have what? 3 carbons plus what? Phosphate each, isn't it?
Yes, Doc.
Yes, so one of the carbons, G, stands for what? Glyceraldehyde. Three phosphate. And this is what? Dihydroxy acetone DHAP, isn't it? Dihydroxy acetone phosphate, isn't it?
Yes, Doc. Yes, Doc.
Then one of them will be converted into the other. Who will be converted to who? One of them, you see these two guys, one of them will be converted into the other. Who will be converted to who?
So I think
Yes.
the dihydroxy acetone
Yeah.
will be converted.
So this one will be converted to this, right?
Yes, Doc.
Said that now we have 2 molecules of what? Glyceraldehyde 3-phosphate. Is that clear?
Yes, Doc.
Gideon, is that
Yes,
clear?
Doc.
Yes, Doc.
Remy?
Yes, Doc.
So this one will be converted. So now you have what? 2 molecules of glyceraldehyde 3-phosphate. Then it will be converted to what? Yes.
1,3 bis phosphoglycerate.
Good. Or di. Base is the same as di, okay? So base phospho base phospho. And I actually made us go through this because of the 2,3 base phosphoglycerate that is needed in the oxygen hemoglobin dissociation curve. Do you remember? That's why I made us go through all these. But ideally, you're not supposed to know all these giddy, but I mean, you can't predict them, okay? Remember that in red blood cells, this one can actually, the positions of the carbon, the one can be moved to 2, okay? To form what? 2,3 diphosphoglycerate. And that is what the red cell used to what? Regulate how its hemoglobin will bind to oxygen tightly or not. Is that okay?
Yes, Doc.
Good. Then it will eventually be converted to what? 3-phosphoglycerate. The 3-phosphoglycerate. Now we will move the carbon to the second carbon from the third to the second. That's what? 2-phosphoglycerate. Samantha, are you following me so far?
Yes, please.
Okay. Then the 2-phosphoglycerate will be converted to what? Phosphoenol pyruvate, which will ultimately be converted to what? Pyruvate. So the point here is that you need to know the enzyme here, okay? That is hexokinase in most cells, but in the liver, it is glucokinase. In the liver, it is what? Glucokinase. But in most other cells, it's what? Hexokinase, the enzyme that converts the glucose to what? Glucose 6-phosphate. And you need to realize that the rate determining step, okay, the rate determining step of this whole process is actually here. NASMA, isn't it?
Yes, Doc.
Which enzyme is responsible for converting fructose 6-phosphate to fructose 1,6-bisphosphate?
Phosphofructokinase.
Phospho what?
Phosphofructokinase.
You're not done. Kinase 1. Remember that there are two.
Yeah.
They have phosphofructokinase 1, phosphofructokinase 2, isn't it?
Yes, Doc.
So that is a PFK1. And that is the rate determining step for glycolysis. Once this enzyme speeds up, the rate determining enzyme for glycolysis is phosphofructokinase 1. The rate determining enzyme for glycolysis, I repeat, is phosphofructokinase 1, PFK1, and it acts here. So it means that then the rate determining step for glycolysis is what? The conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. Is that clear?
Yes, yes.
So you don't have to know all the enzymes.
Yes.
No. But at least I generated this mnemonic for you to be able to appreciate the steps. But know hexokinase, know this, and at least the steps. Okay, that is fine. So what is the summary? The summary is that glycolysis, okay? The summary is that glycolysis actually picks up glucose and then uses 2 ATP in the metabolism only to what? Produce what? The end product is what? Will be 2 molecules of pyruvate, because remember that each glyceraldehyde 3-phosphate would actually go through the steps, isn't it? So in the end, you have 2 molecules of pyruvate plus 4 molecules of what? ATP. It means that the process itself uses 2 molecules of ATP, and then it produces what? 4 molecules of ATP. So in the end, how many molecules of ATP is produced by glycolysis? 2 ATPs. Okay, so please note that glycolysis actually produces what? 1 molecule of glucose is metabolized to produce 2 pyruvate molecules plus 2 ATPs. Please. Net ATPs, I mean, it's 4, but because it has used up 2 to metabolize the glucose, it means that the net one is what? 4, isn't it? So that's what I want you to keep in your head. Glycolysis, the aim is to what? Produce pyruvate plus ATPs. Is that okay?
Yes, Doc.
Any questions? Any questions?
Doc,
Yes.
so how about the remaining 36? Where would they come from?
It will come from the Krebs cycle and then the electron transport chain.
Okay.
That's why we are seeing that whenever you see that the glycolysis, okay, is actually what? An anaerobic. You see oxygen here. You see that there's no oxygen here.
Yes, Doc.
So the glycolysis is actually an anaerobic respiration. It doesn't use oxygen. So it means that it's less than 36. So the rest of the 36 will actually be generated by the Krebs cycle and then the oxidative phosphorylation. Because the glycolysis is the first part of glucose metabolism. Then the pyruvate will enter the Krebs cycle as acetyl-CoA, and then will generate NADH, FADH, and then those are electron carriers. They will go through the electron transport, and finally oxidative phosphorylation will just yield. Is that okay?
Yes, Doc.
Okay. Can I move on?
Yes, Doc.
Okay. Yeah. So good. So now, the pyruvate that is produced from glycolysis, it is converted to what? Acetyl-CoA, and that acetyl-CoA is what is going to what? Enter into the Krebs cycle. Do you see that?
Yes.
What is the name of the enzyme? I think I've mentioned it before. Which enzyme converts the pyruvate to acetyl-CoA? Pyruvate what?
Kinase.
Pyruvate dehydrogenase. I think I've mentioned it. Pyruvate dehydrogenase. Pyruvate dehydrogenase. So when the acetyl-CoA is formed, then it goes into the Krebs cycle. Do you see that? So now we have ended the chapter on what? Glycolysis. So the next thing is what? The Krebs cycle. Okay. So again, just like I did for acetyl glycolysis, I'll not really bore you with. I just want to focus on the clinical aspects. So the Krebs cycle was right from SHS. All of us know that we didn't like Krebs cycle. The Krebs cycle is sometimes called the, I think, tricarboxylic acid cycle, the TCA cycle, or the citric acid cycle, okay? You need to understand that the Krebs cycle is actually the major energy producing pathway. The major energy producing pathway in the body. Please note that it is the major energy producing pathway in the body. Major energy producing pathway. Once I'm feeling sleepy now, I have to mention the thing over and over before I remember what I'm saying. Major energy producing pathway in the body. And where does it occur? Where does it occur?
Mito A.
Come again, mito A.
Mitochondria.
Mitochondria.
Occurs in the mitochondria. Okay. So how is fuel sent to the Krebs cycle? How do we make the fuel so that it can enter the Krebs cycle? Fuel is fed into the cycle as what? Acetyl-CoA. So if you are a fuel, if you want us to generate energy from you, you have to be converted to what? Acetyl-CoA. That is why the pyruvate didn't enter the Krebs cycle as what? Pyruvate, but acetyl-CoA. It had to be turned into acetyl-CoA. Is that okay?
Yes.
Now the cycle, let me just show you a picture of the cycle. Very annoying. But because of the basic sciences, I have to make sure that I equip you with that so that you don't go there and then you are found wanting. Okay. Let me try downloading this. I just want to make a point. Okay. This web file. Let me pause the recording. Can you see, please?
Yes, Doc.
Yes, Doc.
Good. So do you see that the pyruvate is what? Do you see the pyruvate here? from the glycolysis, right?
Yes, Doc.
So that it is converted to acetyl-CoA by what? Pyruvate dehydrogenase. You see that? And acetyl-CoA would enter the cycle. So the Krebs cycle actually begins here. It's a cycle, okay? So it's in the mitochondrial. I said treadmill, no? Like that. So if you want energy produced, just add what? Acetyl-CoA, and then it will generate energy from that acetyl-CoA you have brought. Okay. So it is like Nika. You know Nika? I don't know the English word for Nika. You know Nika?
Yes, Doc.
Hey, who responded with
Granny milk.
why are there grinding milk and a grind milk? Who say almost a machine now? So it is actually going wrong. So if you want something to be grinded or grounded, I don't know, you just put it inside, it goes through and it comes out and it gives you your end product, isn't it?
Yes, Doc.
So the Krebs cycle is just like Nika. So it begins here. Oxaloacetate is the molecule the Krebs cycle starts with. What did I say?
Oxaloacetate.
Is the
Oxaloacetate is the molecule that Kreb cycles. That's right.
Oxaloacetate is the molecule I could have taken the pain to go through all these, but Charlie, yeah, you see that. So the next thing after that is citrate. Then from there, isocitrate. Then alpha ketoglutarate. Then succinyl-CoA. Then succinate. Then fumarate. Then malate. Then anamenye mamona. Send me glycolysis no mnemonic mamona. Let's see. O C I alpha A S S F M OCI ASSFM. Okay, so let me pause there. Yes, so please, this mnemonic is what I generated for the the components of the Krebs cycle, okay, the TCA cycle. So I was saying that the TCA cycle begins with oxaloacetate, okay, and then it goes through some components, but we're trying to actually get a mnemonic so that we can remember the components very fast. So oxaloacetate, so once caught in a strange situation, follow the Messiah. So follow the Messiah. So oxaloacetate, citrate, isocitrate, alpha ketoglutarate, succinyl-CoA, succinate, fumarate, and then malate. Okay, so we want to, please hold on. All right, great. So let's go back to that picture again. So yes, so it begins here. As you can see, it actually begins here. So what happens? The acetyl-CoA is actually added to what? The oxaloacetate to produce the what? Citrate. You see that? That's why we are saying that the oxaloacetate is what? The parent molecule, okay? Is that okay? So it is true that here, acetyl-CoA is actually fed into the cycle, but so for the cycle to continue, the acetyl-CoA has to bind acetyl-CoA has to bind to what? The oxaloacetate to produce a citrate, which produce isocitrate, alpha ketoglutarate, succinyl-CoA, like that. Is that okay?
Yes, so.
Now, what other point do I need to make here, apart from the ones I've made? I want to tell you that, listen, as the acetyl A, moves through the cycle, okay, it actually gives some electrons to some electron carriers, and one of them is this guy. You see that? NAD+. Do you see that? Hello?
Yes, sir.
Good. And then what else? There's another guy. I'm looking for him. I don't think he's here. I'm looking for him. FAD2. He should be somewhere here. I don't know why I can't find him here, but yes, the whole point is that the Krebs cycle actually produces two groups of electron carriers. Remember that the electrons will go and move through the what? Will carry electrons through the electron transport chain, and then that electron will finally get to what? Oxygen, and there's oxygen that will convert everything to ATP. Do you understand me? If you don't understand me, tell me. I will explain myself again. Do you understand me?
Yes, sir.
Okay. So the whole point is that after you've gotten this, please note that the outcome is that you are going to give the energy in the form of what? Electrons to electron carriers. So the Krebs cycle actually produces two groups of electron carriers, okay? Two groups of electron carriers. Those are NAD or NADH and then FADH. So these electron carriers actually come out of the Krebs cycle, okay? Now, the NES stands for, I think, nicotinamide. If I'm not, I'll check for you right now. Nicotinamide something. Please, let me confirm for you. Because I want to make a statement. That's why I'm bringing this up. So the NAD stands for nicotinamide adenine dinucleotide. Please don't keep this in your head. You don't have to keep this in your head, but I don't know. Sometimes I have the feeling that they might just NADA stands for as an MCQ question. Very, very funny. But why did I bring this up? I want to tell you that what is actually needed to produce this is actually niacin. What is niacin? What is niacin? Hey, answer. I've taught you.
Vitamin B3.
Exactly. Vitamin B3.
Vitamin B3.
Exactly. That's true. Vitamin B3. We see that vitamin B3 is needed to produce this, right? So we need it in the Krebs cycle and also in glycolysis, isn't it? FADH2 is called flavin adenine dinucleotide. When you hear of flavin, which vitamin comes into your head?
B2.
What? Be confident. Be confident.
Vitamin B2.
Yes, because vitamin B2 is what? Ribo or riboflavin, isn't it? So please take note of the vitamins that are needed. Vitamin B2 and vitamin B3. Take note of that. Vitamins required in the TCA cycle. Vitamin B3, vitamin B2. There are others, but I'll talk about them later. But the point is that these guys are what? The nicotinamide adenine dinucleotide, okay? They are, and then the flavin adenine dinucleotide. They are actually electron carriers that can what? Eventually send the electrons through the electron transport chain. So the electron transport chain is actually a chain of what? Carriers, which receives electrons and eventually will give it to what? Oxygen. And the oxygen will what? Do the oxidative phosphorylation. Is that okay? Did you get that?
Yes, Doc.
Good. So the point is that Na, I want to make sure that you get it, and then once and for all, biochemistry, although I have protein metabolism and fat metabolism to do, this one you have it engraved. They are produced by the cycle. They are produced by the cycle, by the TCA cycle. And they are actually what? They are electron carriers. Which would give the electrons they have picked from the TCA cycle to what? The electron transport chain. All of you have heard of that before, right?
Yes, Doc.
Now, what is the total energy generated by one round of the cycle? Let's say if you put acetyl-CoA inside one round, it goes and counts like this. How much energy do you generate? You generate 10 ATPs. The total energy generated by one round of the cycle, okay? So let's say we have oxalo. Now that we know the components, now we want to do ISM, isn't it? Jaru. So oxalo acetate. Once hot, right? In a strange situation follow the Messiah. So we add the acetyl-CoA here, isn't it? Then the cycle goes. So I say that when one molecule of acetyl-CoA is added, the cycle goes, one cycle, bang. What we produce is what? 10 ATPs. If we bring, remember that the pyruvate is what? 2. And then you can convert 2 pyruvates into 2 acetyl-CoA molecules. So that means that we can generate about 20 ATPs from 2 pyruvates, isn't it?
Yes, Doc.
So the total energy generated by one round of the cycle, starting with 1 mole of or 1 molecule of acetyl-CoA, will generate 10 ATPs. Now let's see something. I mean, they are all electron transporters, right? How much energy does NADH generate after it generates 2.5 moles of ATP? Please note that I've been asked this question in level 200 before. And then FADH2, this one will actually generate just 1.5 moles, okay? Whenever they produce, and then you see that, okay, 1 molecule of NADH go through the chain, generates 2.5. You go through the chain, also generates 1.5. Is that okay?
Yes, Doc.
Yes.
Now I must say that the enzyme that works here, I hope the thing is not boring.
No, Doc.
Good. So the enzyme that works here, okay, you see that the enzyme that converts alpha ketoglutarate, alpha ketoglutarate to succinyl-CoA. The alpha ketoglutarate dehydrogenase, okay, it needs NADH. It actually needs FADH as well, and it needs vitamin B1. Please note that. Thiamine. So vitamin B1 is also one of the vitamins that is needed in the Krebs cycle. So you've seen B2, also B1, B2, B3. There are others, but let's stick to this alone, okay?
Yes, Doc.
So now that is a Krebs cycle. So I'm done with the Krebs cycle. That is all I think you need to know. So let's go to the electron transport chain. So actually ATP is generated when the electrons from the carriers NADH and FADH are able to pass to what? The molecular oxygen. But the point is that how do they get through to the molecular oxygen? How? They have to actually use a transport system, and that is called the electron transport chain, okay? So the NADH, the FADH2 would have to use a transporter or a transport system. That is called the electron transport chain, ETC. So what are the components of the electron transport chain? So we have the NADH dehydrogenase. NADH dehydrogenase. So it's actually a complex. I think let me look for a picture, okay, so that it will be clear in your heads, okay. Let me see electron transport chain diagram. Electron transport chain diagram. Yeah. Sharp. Oh, nice. Let's see. Oh, nice. Good. ETC. So download it. Good. So can you appreciate it?
Yes, Doc.
So yeah, so remember that the Krebs cycle also takes place in the mitochondria, okay? And it has produced some electron carriers, okay? And you know the two of them, FADH2 and then NADH. So this is the electron transport chain. So you have to actually move the electron throughout till we get to oxygen, and oxygen will do the oxidative phosphorylation. Okay, so this is the first complex, second complex, third complex, and fourth complex with some guys in between them. Okay. Is that okay? So the first complex is called NADH dehydrogenase complex. This one, complex 1. Is that okay? Please hold on.
Yes.
That is complex 1. So complex 1 is actually what? NADH dehydrogenase. Okay. Are we good?
Yes, Doc.
Okay.
Yes.
Good. And then we have the second complex. Okay, I think that is the please let me confirm. It's been a while. Hold on. Yes, so the electron carriers, as you can see, you see them going through the transfer system. Do you see that? So NADH goes, releases its electron, FADH goes, releases its electron. Do you see that? And then they'll go through the various complex. You have complex 1, complex 2, complex 3. I wanted to say sorry, complex 4. And then these will take up the electrons and then give it to what? Oxygen at the end. And the oxygen will do what? Oxidative phosphorylation and will generate the ATPs. That is all. Do you see that?
Yes, Doc.
Okay. Now listen, some clinical correlations, a few of them. Now, you see cyanide, eh? Cyanide, when someone has cyanide poisoning. When someone has cyanide poisoning, or carbon monoxide poisoning, these guys, okay, carbon monoxide, cyanide, actually block the fourth complex. When they bind to it, they block it, so they prevent what? The transport, because that is the ultimate one that will give the electron to the oxygen. So they block the release or the transport of what? Electrons finally to the, so you see that when someone has cyanide poisoning, the ATP actually comes down because what? The electrons were not completely transferred to oxygen, and so we can't produce the ATP. Do you understand?
Yes, Doc.
Do you understand? Good.
Yes.
So what is the summary here? The summary you are to keep in your head for clinical exam is that glucose metabolism begins with glycolysis. And then God being so good, you know the steps. Then it produces what? 2 molecules of pyruvate. And ideally, 4 molecules of ATP. But remember that when we're doing the process, and we use 2 to help in the process. So the net would be what? 2. Then the pyruvate is actually converted to what? Acetyl-CoA by pyruvate dehydrogenase. So the acetyl-CoA is the one which is going to enter the Krebs cycle. It binds to oxaloacetate, which is the molecule that begins the Krebs cycle. Oxaloacetate will actually move to what? Citrate like that, and then the cycle moves on. And then in the end, what you are going to generate is NADH and then FADH, which are electron carriers. Then they will go through the electron transport chain, the complex 1, complex 2, complex 3, complex 4, with some intermediaries, okay? Like the kill, like the cytochrome and all that. You don't need to know that. So the point is that these guys would send their electrons. So this one will use the first complex, this one will use the second complex. And eventually oxygen will be at the receiving end. Okay. And then once it gets the oxygen, it gets the electron, it phosphorylates the electron. So that process of phosphorylating the received electrons is what is called oxidative phosphorylation. And that is what will release the what? Energy in the form of ATP. Is that okay?
Yes, Doc.
Any questions? Okay, so I'm going to put this in the biochemistry portal, okay, as glucose glycolysis, Krebs cycle, oxidative phosphorylation, simplified, okay? All right, please give me a minute. Bye-bye.